Hello everyone, this is the second part of Molecular Genetics Lecture 2, Mutations. In this video, we're going to talk about the different types of mutations and the effect of each type of mutation. The first one that we're going to start with is called point mutation. If you can remember our definition of a mutation, it is a change in the DNA nucleotide due to some sort of error during either DNA replication or the transcription process. So a point mutation is the same as when we say substitutions. It's the same thing. But basically what's happening is we have a DNA nucleotide, which is one of the A, T, C, G letters, that's being switched into a different letter. But remember, the letter is just to tell you the different types of nucleotide, but in real life, the DNA nucleotides don't actually look like letters. They're different, they're actual different types of letters, just like pens. They're all pens, but they're different types of pens. But there are only 4 pens in your DNA. There are 4 types of DNA nucleotides. Anyway, so with a point mutation, what happens is there's one DNA nucleotide base that's being changed into whatever it's not supposed to be in the first place. But the point is, this is only one point in the DNA that's being changed. There's only one nucleotide that's being switched. So here's our definition for a point mutation: change in a single nucleotide in the DNA, usually less serious than a chromosomal alteration. So we haven't talked about chromosomal alteration. We'll get to that later in the video. But for right now, just know that we're changing one DNA nucleotide. And when that happens in your cell, this is called a point mutation. So if you look at this picture right here, we have CTGGAG. That is our original DNA sequence of one of the DNA strands. Obviously, if you change the DNA nucleotide of one strand, when that DNA is replicated, the complementary strand will also be changed. So in this case, the A switched into a G through some sort of DNA replication error, and that is not being caught during the DNA replication proofreading process. So now we got a mutation, a point mutation, to be specific. So these mutations only affect one codon. So if you think about the definition of a codon, it's 3 DNA nucleotides, right? So when the 3 RNA nucleotides, actually, but the RNA is made from a DNA. So when a DNA base is changed, the corresponding mRNA base is also going to be changed. So instead of we would have GACCUC, which is our mRNA, what it's supposed to be in the first place. But with this mutation, you would have GACCCC as our mRNA sequence. So then the corresponding 2 amino acids, because each amino acid is coded by 3 RNA bases, then the second nucleotide that's being coded for is wrong. So if you look at the second part, since more than one mRNA codon can be coded for the same amino acid, sometimes these don't change the sequence at all. So we're going to take a look at the different types of point mutation. There are 3 types. One is called silent mutation or silent mutation. There's a missense mutation and there's a nonsense mutation. And we're going to spend some time looking at each one of them. The first one that we're going to start with is our nucleotide RNA codon chart. So what you're reading right here, each one of them is a codon, and a codon is 3 bases of an RNA that the ribosome can read and translate that information into amino acid sequences. So if you were to have, let's say, here's an example, let's pretend that each one of these is an RNA base or DNA base, but the DNA is going to be transcribed into RNA. So each one of these will code into an amino acid, and since we have 9 RNA bases in total, there should be 3 different amino acids being coded for. So same idea right here. If you have 3 RNA bases, that will code for 1 amino acid. In this case, CCU will code for pro. So in this video, we're going to talk about some examples using English letters, because that may or may not make more sense to you. And if it doesn't, as long as you understand how everything works, it's fine. But if this makes sense to you, go ahead. I'm going to keep on using this chart right here. So we have something called a pseudocode. So instead of having let's say CCU coding for an amino acid LEU, we'll represent LU, an amino acid, with a letter. So let's see. We have C is our first base, right? We read from the left, C, and then we find our second base from across, so CU. So this J, letter J, represents an amino acid that was supposed to be an amino acid, but right here we pretend that this amino acid is a letter so that we can spell out some words. So CCU gives you an amino acid J. So hopefully you understand how to do this. Let's say we have AAU. We have our first base on the left side, A, and then you look across the second base, A and AA, and the third base is on the right side. AAU codes for a small letter S. So you can actually, you can use this to write some secret codes to your friend if you would like to do that. But basically what we're going to do is that for each type of mutation, you're going to do the translation of the original message, which is presented to you as an mRNA, and you'll translate the message with, you will translate that message into a word, and then you'll see what that message is being turned into with a particular type of mutation, and then you're going to highlight the differences between the original message and the translated message, and then we're going to think about what is the actual effect of the mutation. And the effect we're talking about, if this were to be a protein, what would have been changed? So just a quick reminder, we talked about this before, how the DNA, how the amino acid sequence is kind of like a paper, right? There are different types of paper that can, we have a paper that's this big, and we can have a paper that's this big and it's blue. But whenever you have an amino acid sequence, that provides the instruction on how this protein is going to fold. Let's say it's like folding a paper into an origami right here. So the instruction on how to fold that protein is going to give the protein a particular shape that allows the protein to function. Let's say we have this paper. The instruction is already on here. You can't see it, but it's magic. It's on here. And this paper right here is going to automatically fold into this. But let's say there's a mutation on this paper that gets rid of part of it. This is not going to fold into this anymore, which means this protein is not going to function. I understand that this might be a kind of a concept that's hard for you to imagine, but we'll go over that again a little bit on Friday so that it makes a little more sense, hopefully. So let's take a look at the first one. If you're a little confused, that's all right. We'll get there. We'll get there together. Okay. So the first one that we're going to talk about is this message right here. So we're going to take a look at our. Take a look at this chart right here. So with this, our original message, AUG. Let's look, AUG. So I want you to do this on your own. I'm not going to do every one of this with you because it's going to take forever. It's our start codon, so we're just going to write start, or you can just not write start. That's fine. But that's where our message starts. That's great. That's what happens in your cell with making of a protein. Now we can look for the next one. CCG. CCG is L. So our first letter is a capitalized L. I'm going to write this down because I'm going to forget. So you should write this down on the side of your paper as well. So the first one is big L, and then the next one is UUC. So UUC, if you look on the chart, UUC should be A. So our second letter is A, and then UUC, and the next one should be AAA. AAA, if you look right here, AAA is T. The next letter is T. So we're pretending that instead of coding amino acid sequences, we're pretending that we're making a word, right? So just like a protein that needs to be that have amino acid sequences in a certain order, or a certain order of amino acid sequences so that the protein can actually fold and function, an English word also needs to have the sequence of letters that allow the word to actually make sense, right? So it's kind of the same idea. That's why we're doing this exercise. All right, so the next one is AUC. AUC, that's E, letter E, and then the next one is CAA. CAA the next letter should be R. So if you are writing this down with me, as you should be, the word that you came up with is later. So let's see what our last one is. All right, the last one is UGA. So let's look at UGA is a stop codon. So it's a all right, so we have later and a dot because stop codon tells you where the translation of amino acid should stop. So we're also going to stop doing our word. So now we're going to look at the effect of a mutation. So if you scan through this sequence of mRNA nucleotides, you're going to see that there is a change right here at the AGA site. So the AUG is fine, CCG, so our letter big L is fine, UUC, A is fine, and then the next one is AGA. So if you look on this chart, AGA, wait, no, that is, so CC, AGA, that's correct. So AGA, even though we changed an RNA base, AGA is also the letter T, so LATER. So what you find out is that even though we changed an mRNA nucleotide or mRNA base, there's no effect of this mutation. When we had our original word later dot, we now still have our word later dot. The reason for this is because if you look on an earlier page, which is right here, you can see that a lot of the times you would have multiple types of codons coding for the same amino acid, the same codons coding for amino acid. So if there is a mutation on the last base of the third base's codon, then there may or may not be a change of the actual amino acid being coded for. So here's the point. We're getting there. Okay, so if you look at this is what we call a silent mutation, because even with a change in the RNA base or RNA nucleotide, there is no change in the actual amino acid that the RNA codon codes for. So this point mutation is called a silent mutation. So the silent mutation is a subcategory, one possible subcategory of a point mutation. So now you need to think about the effect of the mutations, because there are many different types of mutations and many different types of effects that may or may not be big. So in this case, what is the effect of this mutation? There's none. There is no effect, right? Because there's no change in the amino acid. So if this had been an mRNA coding for an actual protein, coding for the sequence of amino acid that will allow the amino acids or the polypeptide chain to actually fold into a protein, is there anything that would be changed? The result is there would be no change in the protein overall. All right. So moving on, we're going to look at the same idea. So I'm going to ask you to figure out what this word is with the partner next to you using this chart right here. So pause the video. I'm also going to pause my video and figure out what this word is. Okay. Quick march break. All right, now back to work. So if you actually follow my instruction and wrote down these words on your paper, which you're supposed to, you'll find out that the original message right here says HALLO with exclamation point, and our mutated message was this one mutation word or mutation. Right here, we changed this U to an A. This is a point mutation. It spells HELLO with an exclamation point. So as you can see, this is a point mutation just like the last example that we saw. However, we actually changed the amino acid that the mRNA nucleotides are coding for. So if you take a look right here, this is called a missense mutation. So a silent mutation is silent because it's not changing the amino acid, so it's not changing the protein. A missense mutation is when there's a change in the original DNA, which changes the RNA bases, right? Because the RNA is made from the DNA. So our mRNA actually changes the nucleotide that is coding for. So In this case, the effect of this mutation is one letter was changed. You see one letter, as in the nucleotide letter, not the A and U letter. And if this mRNA were to be a protein, what would have been changed? One amino acid will be changed. This is called a missense mutation. That's the definition. All right, our next one, we have another message. Go ahead and translate this message using our chart right here. Pause the video, figure it out, and then figure out our mutated message as well. So now you've done both of those, and you can see that something else happened. So instead of having a silent mutation where there is no change in the amino acid, or a missense mutation where there is a change in the amino acid, we have something called a nonsense mutation, where this amino acid change actually, instead of changing to a different amino acid, we got a stop codon before the stop codon is supposed to show up. So what happens is, let's say your nucleotide mRNA nucleotide is supposed to code for 15 amino acids that will, be a part of the protein. Actually, it's usually way longer than that. Let's say it was supposed to be 15, but a premature stop codon is introduced so that your polypeptide chain is now only 5 amino acids long. So instead of our polypeptide being this long, now it's this long. So if your polypeptide chain is really short, or even if it's shortened by a little bit at all, this is going to be a huge problem because our protein, this is not going to work. There's not enough material to make a protein. So let's say this is our original paper, right? Oh, that's the wrong paper. So let's say this is the paper that we're trying to fold into this origami. However, a premature stop codon is introduced that now our paper or our amino acid chain is only this long. This is not going to form the correct protein, right? So what is the effect of this mutation? The message stopped prematurely, and if this had been an mRNA coding for a protein, what would have been changed? Only a part of the protein is synthesized, right? So you will have shortened polypeptide chain or a shortened amino acid chain, and the result is the protein is most likely not going to function correctly. So if you think about our silent mutation, missense mutation, and nonsense mutation, all those three types of mutations fall under the bigger category of a point mutation, where a change in one nucleotide introduces, some sort of effect. So a quick review from the previous lesson. Just take a look right here. All right, so if you look right here, this is showing you how you would always read an mRNA sequence. This is an mRNA sequence, 3 bases at a time, 3 bases at a time, 3 bases at a time. So if you look right here, there are 3 types of substitution. This is what you actually write in the notes. There's a silent mutation that allows a mutated codon to code for the same amino acid, but you're changing one base in all these different types of mutations, point mutations. And for example, you can have CAA glutamine, you have a change in the last base, and now you have CAG as your codon, but it's still coding for a glutamine, which is an amino acid. So what is the effect? There is no effect. Your protein is just fine. Everything in your body will function just fine. How about missense mutation? Missense mutation is when a mutated codon codes for a different amino acid. So for example, instead of CAA, which codes for the amino acid glutamine, you can change the second base into a C, and now CCA codes for proline, which is a different amino acid. The effect of this, sometimes this change in one amino acid might affect how the protein is folded. So instead of, our functional protein, you might have something that looks just like this, possibly. Or you might have a protein that's misfolded because of this changing amino acid. That's also a possibility, right? So the effect is variable. However, the most intense one, right here, is called nonsense mutation, where a premature stop codon, premature means your stop codon is supposed to be right here, but now it's over here, so now your amino acid chain is shortened. That's called a nonsense mutation. The example could be CAA. We can change, not we can change, but a mutation changes the first base into a U. So now you have UAA. This UAA will code for a stop codon. And the effect is usually very serious because your amino acid change is so short. Here's another example. Missense mutation. Let's say your mRNA is supposed to read, or these are the words that's supposed to be there. The cat ate the rat. You change a letter and then it changes the word as well. The cat ate the hat. This is fine, I guess. There's a little bit of change, but not huge. There's one change in amino acid. One change of one change of nucleotide, or point mutation, is change in one nucleotide causing the change in one amino acid. That's our missense mutation. Nonsense mutation, the cat ate the rat versus the cat ate. So now you have a premature stop codon, and the sentence doesn't make nearly as much sense. Now here's an example. Here's a real-life example of a substitution or a point mutation. Sickle cell anemia. So sickle cell anemia is the result of sickle cell anemia is a person with this disease will have red blood cells that kind of shaped like a moon and is not nearly as good as carrying oxygen as a regular red blood cell. So the result is, if your body isn't getting enough oxygen, things are not very great. So sickle cell anemia, having this misshaped red blood cell, is actually caused by changing only one base. So what happens is the GAG codon is changed into GTG. Wait, but this is not a codon. This is your original DNA. GAG is changed into GTG, which eventually leads to the change in amino acid from glutamine to valine. And this only one change in one amino acid out of all the amino acids out there makes the hemoglobin, which is the protein that helps carrying oxygen in your body, in the red blood cell, it causes it to misfold. So now your protein is misfolded, and now your red blood cell is not as good at carrying oxygen, and that's pretty terrible. So if you think about it, this is one small error causing a lot of big problems, which is entirely possible. Feel lucky that you don't have a lot of problems. So we're going to be done with this video. So far, we talk about point mutation. There are 3 types: missense mutation, nonsense mutation, and silent mutation. You need to think about the first one, that they both fall under the category of point mutation or substitution. You need to think about the effect of it. Sorry, grab it. Silent mutation doesn't change the amino acid. The effect on the overall protein shape, the protein is the same as it was supposed to be. The protein will function just fine. Okay, so when you are doing your homework of making a Quizlet, make sure you're asking yourself these questions in the Quizlet, right? What are the 3 types of point mutations? What is a point mutation? What is the effect of a nonsense mutation, a silent mutation, a missense mutation? Let's go over this one more time. Think with me. We already went over the silent mutation. A missense mutation is a change in one DNA nucleotide causing a change in the codon that changes one amino acid. And the effect may vary. Sometimes it might be, the effect might be really severe. Sometimes it might not cause that many changes. And then the last one is our nonsense mutation, is when a change in one DNA nucleotide causes a change in one codon, which makes the original codon that codes for one amino acid actually codes for a premature stop codon. So now the effect on the overall polypeptide chain is that the polypeptide chain is shortened, and the protein is most likely going to misfold and it won't function. Okay, so kind of summarize all of these ideas, make sure that it makes sense to you in your head. Just one last note on why exactly does the the missense mutation, why does it have different, possibly different effects? So originally, when we talk about the different types of amino acids, we kind of touched on this, that there are different amino acids might have different characteristics. Some might be positively charged, some might be negatively charged, some might have no charge, some are hydrophilic, like they like water, some might be hydrophobic. So if we were to change a hydrophilic amino acid to another type of hydrophilic amino acid, for example, that might not cause that much of a change in the protein that's actually being formed. However, let's say we're changing a hydrophilic amino acid into a hydrophobic amino acid. This is definitely going to change how the protein is going to fold, and this is going to change, the overall shape of the protein, and this protein is most likely not going to function. So both examples are when we're changing one nucleotide, which changes one amino acid, but the effect varies because of the property of the amino acids. So while you're doing your homework, make sure you think through all these different concepts, make sure that you actually understand what it means. Use the homework to help you review these concepts, and make sure that you ask questions on Friday if you have any questions.